about common neurology. I think this information was given on lecture. I think, but I want to repeat this once again because it's very important, and I want that you understand this. And first of all, let's talk about classification of nervous system. Now, who can tell me classification of nervous system? How can I divide this system? Doctor.
Central nervous system,
And peripheral nervous system. If we divide nervous system into two parts, central and peripheral, that's why we must know differences between them. Well, all students of 10th group, I can divide it into two parts. Which one? How can I divide it? Boys and girls, what are differences? Well, who are you, boy or girl? Why? What is the difference between boys and girls? Oh, structure of body, structure of reproductive system. Because no differences between them standard, but differences in reproductive organs. And now, if you divide nervous system into central and peripheral, you have to know differences between them. Which differences between them? According to which feature we divide nervous system into two parts, central and peripheral? On the basis of what? Huh? Localization. How localization is called by scientific language? Localization. First letter is T. T. Second O. Topography. So this topographical division, right? That's why our nervous system is divided on central nervous system and peripheral nervous system, and differences between them is in topography. Logically, central nervous system is in the center. Look, we said central nervous system includes brain and spinal cord, right? Brain in the skull, which is in the center, and spinal cord, where is? What is the spinal cord?
Vertebral canal.
In the vertebral canal, which also in the center. In the center. That's why brain and spinal cord, both of them are localized in the center of our body. That's why it's the central nervous system. Well, tell me please, what is belong to the peripheral nervous system?
Nerves.
Once again?
Plex.
Nerves. Don't
Plex.
need to divide cranial or spinal or just nerves.
Plexus.
Plexus. Once again. What did you say? Ganglion, very good.
Receptors and terminals.
Receptors and others. So everything which is not localized in brain and spinal cord belongs to the peripheral nervous system. Very easy classification. Did you understand this? That's why now our purpose is to study central nervous system at the first, because this base. When I want to investigate Sri Lanka, of course I begin the investigation from capital of Sri Lanka, from Colombo. I don't go to any village there and begin to investigate Sri Lanka from village. I come to the center. Same, when you come in Belarus, of course I think first city which you visit is Minsk, is capital, not Grodno. So same here, we must investigate the central nervous system. Then, who knows second classification?
Somatic and
Somatic and autonomic. Again, if we divide into two, somatic and autonomic, I must know differences between them. What are the differences between these two parts, somatic and autonomic?
anatomical function.
Functions, right? This different functional classification, autonomic and somatic. Autonomic and somatic. So we said functional differences. Well, tell me please. Well, somatic and autonomic. If you said that there is functional differences, tell me please. First, very important to understand that our somatic nervous system is voluntary. What does it mean voluntary? What is the meaning of this? You can control. What can you control in our body? What?
Skeletal muscles.
Skeletal muscles. I want to touch my nose. I contract my muscles and I touch my nose. I don't touch my shoulder or cheek, I touch my nose because I want to touch my nose. Now I want to come to the blackboard and write something. So I contract my muscles and I come here. That's why somatic nervous system, which is voluntary, must innervate what?
Skeletal muscle.
Skeletal muscles, not muscles, but exactly skeletal muscles, because we know there are three types of muscular tissues. Skeletal muscles. Well, something else?
Plants.
Glands. No? What else you can't control or you can feel? What can you feel? Hmm? Do you feel? Touch. So, if you feel that I touch you, that's why what does somatic nervous system control? What does it innervate?
Senses.
No, sensory of what? I didn't touch your stomach.
Sensor of touch. Skin.
Skin, of course, it innervates skin because when your food enters your stomach, do you feel it? Do you feel this? No, you swallow something and food passes through the esophagus. Do you feel this passage? That food irritates mucus of your esophagus. Do you feel this? No. When food enters stomach, do you feel this? No. That's why this is not somatic nervous system. But when I touch your skin, I touch your hand, do you feel this? Yes, of course. If in bus somebody touch you, do you feel this? Of course we feel. That's why somatic nervous system innervates just two parts, skeletal muscles and skin. Only these two structures. Understood? Or not? Understood. Well, autonomic nervous system, what does it innervate? What is according to this? Which is the autonomic nervous system?
Involuntary.
Involuntary, of course. Involuntary. Involuntary means you cannot control it. It is autonomic nervous system. Autonomic. Right? It can work itself without our control. So tell me please, what does this autonomic nervous system innervate?
Cardiac muscles.
Cardiac muscle, this specific muscle. Cardiac muscle, okay, then.
Smooth muscle.
Smooth muscles, where can I find smooth muscles?
Organs.
In
Internal
organs, that's why internal organs, all internal organs. But do you remember, you already passed splanchnology, the anatomy of internal organs, which organs have both muscles, smooth muscles and skeletal muscles?
Stomach.
Stomach, really? You can control your stomach?
No.
You enter, you swallow something, then you think, this food was bad, I don't want to digest this food. Can you vomit this food? No, you cannot. Itself, this involuntary process, vomiting.
Urinary bladder.
No.
Urinary bladder, no, urinary bladder is involuntary. You cannot dilate your urinary bladder. Rectum, of course, distal part of rectum, canalis analis, has two sphincters. One is internal sphincter, another external. You remember that external is voluntary, internal is involuntary. Well,
Esophagus.
esophagus, upper part of esophagus is voluntary, skeletal, you can't control because upper part of esophagus participate in swallowing. You put your food in the mouth, you chew, can you decide, can you swallow this food or not? Of course, I can chew, but I cannot, I don't want to swallow. And this food go out, right? But sometimes, no, I will swallow better. And you swallow. This voluntary process. That's why upper part of esophagus consists of skeletal muscles, lower of smooth muscles. That's why, for example, our esophagus or our rectum is innervated by both somatic and autonomic nervous system because it has two types of muscles. Understood this? The next one.
Glands. And
Glands, of course. Glands. For example, sweat glands, because pancreas, liver, which are glands, belong to the internal organs. Are glands which are in skin. Right? For example, sweat glands, sebaceous glands, and other. Lacrimal gland, and other. Well, and blood vessels. Blood vessels or lymphatic vessels, any vessels. So, that is why, look, our autonomic nervous system is widely spread. is larger systems than somatic, because somatic only skeletal muscles and skin, but autonomic, look how many structures it supplies. Understood this? Did you understand this classification or not?
Yes.
Yes, you understand. Do you have questions about this? That's why now you must understand these differences, because on our final exam, everyone will get exam card, and in every exam card, second question is called internal organ, for example, stomach, structure, blood supply, innervation. And you just, if you will talk about innervation, you just think, stomach, can I control the stomach? No. If you cannot control the stomach, that's why you have to use autonomic nervous system for innervation. Then tongue. Your question is tongue, blood supply, innervation. Tongue. Can you control the tongue? Yes, of course. Show me your tongue. So you can't control. That is your wish. That's why which system innervates the tongue? Somatic, of course, because you can't control. Okay, let's talk about muscles of smile, muscles of facial expression. What? Yes. Somatic, because you can control. Please smile. Even if I don't want, but I smile because it's my wish to smile. So understood this. Okay, if you understand this, let's go here. Next, tell me please, what our nervous system consists of from?
Neurons.
Neurons. Neurons are specific nerve cells, and these nerve cells consist of two basic parts. What is the first part?
Center. Body.
Body, or this one, body and process. Just two types, no two parts of any neuron. No more parts. So, according to this structure, look, which types of neurons we can distinguish. First, how do you think? How this type of neuron is called? Which one? Unipolar. This is unipolar. Why unipolar? Because it has one corpus, one body of neuron, and only one process. Then, unipolar. One process starts, but then it divides and goes to the different parts of my body. Next one, multipolar, no, and bipolar. Very important to remember this one, pseudo-unipolar, because very often we'll meet these pseudo-unipolar neurons in our semester, this semester. Well, did you understand this? Understood. Now, tell me please, if I take all bodies, only bodies, what do they form?
Ganglia.
What?
Ganglia.
Only bodies. If I will look only on the bodies. Gray matter. That's why our nervous system consists of two types of matter, two types of substances. If you take a fresh brain or a fresh spinal cord and you cut it, you can see just two colors. So one of them is gray, another white. But look here. Here, look. Gray, and white. So, and now you know that gray matter includes bodies of neurons. So what does white matter include?
Processes.
Processes. So all these processes, they are white matter. So tell me please, do you remember why color is different in bodies and processes? Because of what?
Myelin sheath.
Because of myelin sheath. That our fibers are covered by myelin, and because of this, they look like a white color. Well, if we divide nervous system into two matters, gray and white, we have to know differences between them. Logically, this must be your principle. If you divide something, you must know differences. Hypertonic disease. Hypertonic disease is divided into 4 stages. Why we divide into 4 stages? What are the differences between them? No, maybe because first stage is the easiest. This increasing of blood pressure is, for example, up to 140. second degree from 140 to 160, third from 160 to 180, and the last is higher than 180. Understood this? So tell me please, what are the differences between them, between gray and white matter? What are the differences? Functional differences. There are functional differences. Tell me please, what is the basic function of gray matter, of bodies? What do they do? Why do you need this gray matter? Why do you need these bodies of neurons? For what? Doctor, how do you? Don't know. But this is not new information. You studied this on histology, and you had lectures in previous semester about this. And now you are surprised. What are the functions of gray matter? What are the...
It contains nuclei and the function of it.
Sit down. No need to sit.
Somatic or vegetative function.
Oh, I don't understand. Somatic or vegetative function? What do you mean? What is the meaning of somatic function? What is somatic function? Do you know? Huh? Voluntary function, да? What does mean voluntary function? I don't answer. What does mean voluntary function? You can control what? Movements of the body. You can control. That's why gray matter controls movements of the body. Так? Your heart is moving or not? That's why this gray matter cannot control. Because heart movement is not a voluntary process. Right? That's why heart movement is not under the gray matter control, right? What is written? Analyzer. So why you think? It is written everything. Gray matter, bodies of neurons are necessary for analyzing of information. I touch the table. Without gray matter, I cannot say anything about properties of the table. I touch, nerve impulses come to the gray matter, to the bodies of neurons, and only after that I can understand. Oh, this table is cold, this table is hard, this table is smooth. Because this information is analyzed here. That's why gray matter is very important. Gray matter for analyzing this information. For example, posterior part of the brain, of the hemisphere, is this one, is occipital lobe, which is covered by gray matter. And this gray matter, which is localized here, analyze visual information. I look at you, and I see you boy, you girl, you look at me, I understand this. Why? Because this cortex, this gray matter, analyzes getting information which comes from my eyes. But if I will remove this cortex, I will broke this gray matter, for example, during stroke, during trauma, cranial trauma, and this gray matter is not working. My eyes look, information comes to the center, but center is not working, gray matter is not working. So I am blind. I don't see, I cannot analyze what I am looking, I cannot recognize anything. Understood this? Or not? Yes. Well, what is the function of white matter? White matter, I remember once again, is complex of processes. Why do you need white matter?
Conduct impulse.
Oh, very good. Oh, this you remember. Conducting. Conducting of what?
Impulse.
Of impulses. I touch this table, and to analyze this information, first of all, I must conduct it from the receptors, which are on my skin, up to the bodies of neurons. That's why here, for example, is receptor, this table, and this impulse come from this process up to the body where is analyzing is occur. That's why two differences between gray and white, for analyzing and for conducting. According to this, anesthesiology work. I want to delete my tooth. My tooth, I have tooth pain, I will need to delete it. How? This is very painful. That's why I take anesthetic, any drug, I inject here, not in spinal cord, not in brain, but here in my gum. And what happened? This drug block this nerve which come to my tooth. That's why this receptor receive information, pain occur here, it goes up to this block and cannot pass. That's why my body of neuron cannot analyze this information, and if it cannot analyze, you cannot feel the pain. That's why doctor can delete your tooth and no any pain you feel. Understood this? Understood. Well, now let's continue. Very important moment. I want that you understand this just now. Look, in our bodies there are millions of neurons, and all of them have almost same structure. They have bodies and processes. But some of neurons are localized in one place, and carry out same function. For example, how many students in faculty of the foreign students on the second year? How many?
300.
300
300.
on the second year, 300 students. Okay, 300 students. But only 12 or 11? 11. Only 11 are here in class number 6 in anatomy corpus. Your neuron. So you are bodies of neurons. You are in one place, and now you carried out same function. What are you doing now? You will listen to me and try to think. So how can I call you? Who are you? Group number 10. Because group number 11 is in another classroom. A group number 1 may be in another corpus. And they know any connection between group number 11 and group number 10. So you are group number 10. So if somebody will ask you, who is in sixth class and listen to me? Group number 10. So that's why if body of neurons are localized in one place and carry out same function, for example, what does it mean same function? They control pain. This control pain, this for neurons, they control pain. They are localized in one place, and if this aggregation of bodies of neurons are localized in central nervous system, means in brain or spinal cord, this aggregation is called nucleus. This nucleus. But if this aggregation, which carry out same function, but it is localized in the PNS, peripheral nervous system, so not in brain and spinal cord, but somewhere else, this aggregation is called ganglion. Did you get this?
Yes.
Or not?
Yes.
Yes. So functionally, no differences between nucleus and ganglion, because both of them are gray matter, and both of them for analyzing. Differences is only in topography. Nucleus in CNS and ganglion in PNS. It is great mistake if you said ganglion which is localized in our brain. There are no ganglion in brain, inside of brain. There is ganglion which is near brain, ganglion which is near spinal cord, but not inside. And there are nuclei. There are no nuclei outside. All nuclei, if I said, can you show me where is nucleus ruber? There is nucleus ruber, red nucleus. Of course, nucleus, gray matter, it must be inside of brain. Understood these differences? Please remember, this is very important to know, because up to the end of semester will be used this term, ganglion and nucleus, and you must know what is the difference between them. There are no functional differences, but there is topographical difference. Same we can say about white matter. Say, look, there are a lot of fibers, a lot of processes. But if I have aggregation of fibers which are localized in one place and conduct same impulse, for example, this table, I want to touch this table. I touch this table by only one finger, but on my finger, on this area, which is touched this table, there are a lot of receptors. I don't touch this table by one receptor, but I touch maybe by 100. That's why 100 nerve fibers, 100 processes go to the gray matter to the bodies of neurons, but all of them conduct same impulse, same tactile inputs. Understood this? Or not? Understood or not? Really? So, if complex of fibers are localized in one place under one myelin sheath and conduct same impulse, for example, tactile impulse, and this aggregation is localized in PNS, this aggregation is called nerve. So I touch this table, and with the help of nerves, this impulse, tactile impulse, come to the either nucleus or ganglion for analyzing. But same aggregation of white matter, of fibers, which conduct same function, same impulse, but when it is localized in central nervous system, means inside of spinal cord or brain, it is called tract. So, same like here, there are no tracts outside of brain and spinal cord. Outside are only nerves. When I cut my hand here, amputation of the heart, I can cut what? Tracts or nerves? Nerves only, no tracts here. But when I cut spinal cord, totally fracture of vertebral column fracture, and my spinal cord is ruptured also. What did they rupture? Nerves or tracts?
Tracts.
Tracts. So tracts belong to CNS, nerves to PNS. Did you get this? Please remember this one all your life. This is very important to know. Do you have questions? Oh, no. No questions. I think enough. This is enough information about introduction into neurology. No need to know more. No need to know development. This is not so important for you. You are not pediatrics. That you will not work with children. So that's why this will be enough for you. Now let's talk about what? About spinal cord. Let's talk about spinal cord. So as we already said, spinal cord is in vertebral canal, and spinal cord has this contour. If I cut horizontally spinal cord, look how can you draw spinal cord. Very often you will be draw spinal cord, because even on next class I will give you written test, and you have to draw this schema. On this schema, you have to remember that on your page, everything which is drawn above the higher point of this schema is dorsal. Dorsal or posterior. So if you draw like this, I understand that this posterior part here, which is on the back, faces to the back, as this anterior or ventral. And now you can see differences between anterior and posterior aspects of spinal cord. Anteriorly, just in the center, I can see very deep crack, very deep fissura. This fissura is called fissura mediana anterior. Deep fissura, which is in the center. Posteriorly, on the dorsal side, also depression is visible, but not so deep like in front. That is why it is called sulcus medianus posterior. This feature is very useful to recognize correct position of the spinal cord on the fresh preparations. If you find deep fissura, it means that this anterior surface. Understood this? Okay.
Yes.
Yes. Then when I cut spinal cord horizontally, inside I can see this schema. This picture. What did I draw? Inside of spinal cord, I can see two matters. like we discussed here, gray and white. And you can see that gray matter is in the center of spinal cord, but white matter is on the periphery. Now you know that gray matter is for analyzing. And if we are talking about spinal cord, what can I find in gray matter? Nuclei or ganglion?
Nucleus.
Nuclei, of course, nuclei. And gray matter of spinal cord can be divided into 3 parts. What your group I can divide into 2 parts, boys and girls, but gray matter of spinal cord into 3 parts. First part, this one, is posterior horn, cornu-posterior. This one, lateral horn, and this one, anterior horn. 3 horns. And once again, if we divide gray matter into horns, 3 horns, that's why I must know differences between them. Why did they divide into 3 horns? What are the differences between them? Now I must know this. And now, if you said that in the horns I can find nuclei, now let's talk about classification of nuclei. Gray matter of spinal cord is divided into 3 horns. Logically, there are 3 types of nuclei. And this division of nuclei is according to functions. Nuclei can be, S means sensory nuclei, M
Motor.
motor,
Vegetative.
vegetative or autonomic, that we said. Understood this? That's why now these 3 types of nuclei I can put in 3 horns. How do you think? Posterior horn, which type of nuclei consists? Sensory. All sensory nuclei, you must draw like that. Just ring. So in posterior horn, I can find sensory nuclei, which takes sensitivity. Then in the lateral horn,
Vegetative.
vegetative you have to draw like that. Only like that. Vegetative. No, and logically, which left? Motor. Motor nuclei are localized in anterior horns, and motor you have to draw like that. Did you understand the principle of this? Well, very good. Now, you must know exactly names and functions of all nuclei of spinal cord. Let's begin from the posterior horn. First nucleus, which is localized on the top of posterior horn, is called substantia gelatinosa. Substantia gelatinosa. This one. And you must know function of this nucleus. Function of this nucleus is tactile sensitivity. This sensory nucleus, that's why tactile sensitivity. I touch the table and I feel properties of this table. It is hard, it is smooth, it is cold. So all this I understand because of working of substantia gelatinosa. If I damage this nucleus, I touch this table, I cannot say anything about it. I cannot analyze this information. What we said, analyzing in CNS nucleus, that's why this analyzing of tactile sensitivity is in this nucleus. Next nucleus is in the center of the posterior horn, here. It is called nucleus proprius. Nucleus proprius. Nucleus proprius controls pain. All pain information, pain sensitivity comes to the nucleus proprius. Next nucleus lies at the base of the posterior horn. At the base, it is called nucleus thoracicus or nucleus of Clark. Nucleus of Clark. This nucleus of Clark has brother which lies on the borderline between posterior and lateral horn. It is called nucleus intermedio medialis. Nucleus intermedio medialis. And these 2 nuclei carry out same function. They control proprioceptive sensitivity. Proprioceptive sensitivity. Who remember what is meaning of the sensitivity? What is proprioceptive sensitivity?
Positions of the
Position of
the
body
body.
or recognizing of our body. I want to touch my nose. I know where is my nose. My nose is not here. My nose